All right, let's talk about an organism you might think is just a harmless bystander in our throats, Neisseria subflava. But it has this kind of fascinating, darker side. We're going to get into cases of mistaken identity in the lab, really serious infections caused by medical procedures, and even its secret role in helping create superbugs. We've pulled all of this together using a Gemini deep search, so let's jump right in. And hey, if you really want to get into the weeds and look at the primary research, all the sources we used are right there in the notebook document linked for you. Okay, here's our game plan. First, we'll tackle its two-faced nature. Then we'll hit the lab to see how to properly identify it. After that, we'll look at what happens when it goes from harmless to harmful, how to treat it, and, super important, how to prevent those infections. We'll wrap things up with its pretty worrying role in spreading antibiotic resistance. So first up, we're going to explore this dual identity of Neisseria subflava. Is it just a friendly neighbor, or is it a threat waiting for an opportunity? This is really the central puzzle, isn't it? For most of us, Neisseria subflava is just there. It lives in our throat, doesn't cause any problems at all. But then, something flips a switch, and it can become a pathogen capable of causing some incredibly serious, even life-threatening diseases. The key is figuring out what causes that transformation.
Now, here's where it gets really tricky, and frankly, it's a huge headache for clinical labs. This bug can be mistaken for its notorious cousin, Neisseria meningitis. They're so similar that one of our go-to technologies, MALDI-TOF, can get them confused. And that is not a small mistake. A misidentification can trigger a massive and completely unnecessary public health response.
Okay, so let's head over to the lab bench and figure out how we can reliably tell these two apart and avoid that diagnostic mess. So the first clue we get is under the microscope. We do a gram stain, and just like other Neisseria, we see these little gram-negative diplococci, with that classic flattened coffee bean shape. This tells us we're in the right ballpark, but it definitely doesn't tell us the exact species. So when we culture it, we start to see some key behaviors. It's not too picky. It'll grow on blood and chocolate agar. It's oxidase positive, catalase positive, but a couple things really stand out. First, it gets wiped out by colistin, so it won't grow on selective media like Thayer Martin. And second, it produces this really distinct yellowish pigment. In fact, that's where its name comes from. Subflava means somewhat yellow. Now, to be absolutely sure, we run some biochemical tests. It'll ferment glucose and maltose, just like meningitidis. But the real giveaways are its sucrose reaction, which can be variable, and the fact that its DNA is positive. Those subtle differences are what let us say with confidence, nope, this isn't meningococcus. And why is that so important? Just look at the difference here. A report of meningitis, that triggers alarms, patient isolation, a massive public health response, contact tracing, the whole 9 yards. A correct report of subflava, that means calm, targeted treatment for the patient, and no public health panic. So yeah, getting the ID right has huge real-world consequences. Okay, so let's connect what we see in the lab back to the patient. When and how does this bug go from just a colonizer to a dangerous pathogen? Well, once it gets into places it shouldn't be, like the bloodstream or spinal fluid, it can cause a whole spectrum of really severe diseases. We're talking meningitis, infections of the heart valves, sepsis, and even deep infections in bones and joints. And here is the #1 risk factor. It's not always about the patient having a weak immune system. The biggest trigger is often a medical procedure that breaks through our natural defenses. A healthcare worker who has this bug in their throat can accidentally introduce it into a patient's spinal canal during, say, a lumbar puncture. That leads to what we call iatrogenic meningitis, an infection literally caused by medical treatment. It's a perfect storm. So let's talk about treatment. This part gets complicated because managing these infections presents some pretty unique challenges for doctors. This quote from EUCAST really nails the problem. There are no official breakpoints for the species. That means the lab can't just report and isolate as susceptible or resistant. All they can do is give the doctor the raw number, the MIC, and the doctor has to use their clinical judgment to figure out what to do with it. So, what does the data say? Well, looking at case reports, the clear winners are third-generation cephalosporins, like ceftriaxone or cefotaxime. Forget about penicillin, there's just too much resistance out there now. The length of treatment really depends on where the infection is, with longer courses needed for serious stuff like meningitis and endocarditis. For kids, the story is pretty similar. Third-gen cephalosporins are still the way to go. But there's a really important detail for newborns. Cefotaxime is usually preferred over ceftriaxone. That's to avoid the risk of a serious condition called kernicterus. And of course, with kids, everything is strictly dosed based on their weight.
Now, for the good news, preventing these iatrogenic infections, the ones caused by procedures, is actually pretty straightforward. It all comes down to simple steps that have a massive impact.
The evidence on this is crystal clear. The single most effective thing you can do to prevent this type of meningitis is for the person doing the procedure to wear a simple surgical face mask. Anytime they're accessing the spinal canal, that mask creates a physical barrier. It blocks their own respiratory droplets from contaminating the patient's sterile space. It is such a low-cost, high-impact way to improve patient safety.
All right, last but not least, let's zoom out and look at the bigger public health picture, because this is where Neisseria subflava's story takes another dark turn. This might be the organism's most significant long-term threat. See, Neisseria species are what we call naturally competent. Basically, they're experts at swapping DNA with each other. This turns Neisseria subflava into a kind of lending library for antibiotic resistance genes, which it can then pass along to its more dangerous cousins. And here's the smoking gun. We know for a fact that Neisseria subflava can donate a resistance gene called PEN-A to Neisseria gonorrhea. And what does that do? It makes gonorrhea harder to treat with cephalosporins. So think about that. This supposedly harmless throat bacteria is directly contributing to the rise of drug-resistant gonorrhea, a massive global health threat. This all leaves us with a really tough question for the future, doesn't it? The line between harmless bacteria and resistance reservoir is getting blurrier every day. As these reservoirs grow, how do we have to change our strategies for antibiotic stewardship and infection control? We can't just focus on the pathogen of the day anymore. We have to consider the whole neighborhood. And that's our look into the surprisingly complex world of Neisseria subflava. If you enjoyed this deep dive, please subscribe for more explainers into the amazing world of microbiology. We'd love to hear your thoughts, so definitely leave your questions and comments below. Thanks for watching!